Hello everyone. Welcome to this particular lecture in medical microbiology. And in this particular lecture, we are going to have an introduction to medical microbiology. So without wasting much of our time, let's get straight into this particular lecture. All right, so by way of definition, what is microbiology? So basically, microbiology is just simply the study. Okay, it's just simply the study of tiny living things. So these tiny living things are what we refer to as microorganisms. Okay, so we can say microorganisms or we can say microbes. So these microorganisms um, are living are tiny living things that you can see with your naked eyes. So what that simply means is that you can only get to see these microorganisms basically when you use a microscope. And these particular microorganisms or microbes include you have bacteria. Okay, you have bacteria. So bacteria are just single-celled organisms which can actually live in many environments. Okay, so some of the some of the bacteria can cause disease, while some other kind of bacteria are very helpful. Okay, they can help in digestion or in food production. Then we also have viruses. Okay, so viruses are very small and can only grow inside living cells. Okay, then we also have fungi. Then we also have so on fungi we have fungi can be single celled, for instance yeast, or they can be multicellular such as molds. Okay, so you find that some of the fungi are infectious. Okay, some fungi are infectious. Then we also have protozoa, and then on protozoa, these are simply more like animal-like microbes, okay, which live in water or in moist environments. Okay, then we also have algae. Okay, we also have algae as an example of the microbes that microbiology tends to study. Then that was with regards to the microbes that microbiology focuses to study. We also have branches of microbiology. So these are just some of them. The list has not been exhausted in this particular lecture. Okay, so we have bacteriology. Okay, we have bacteriology. So bacteriology, just from the word bacteria, and logy just means study, then bacteria. So this is just the study of bacteria, and this particular branch of microbiology basically helps doctors to understand infections. Okay, to understand infections, for instance, pneumonia and tuberculosis, which are caused by bacterias. Okay, then we also have virology. Okay, we also have virology, which is just the study of viruses. Then we have mycology. Okay, for mycology, mycology is just the study of fungi. Okay, it is the study of fungi, and it is very pertinent or very important when it comes to treating fungal infections. When it comes to treating fungal infections. Now, it is very important. First of all, before we can even talk, when we talk about fungal infections, what are these fungal infections? Because there is where most students tend to confuse or to think that fungal infections are the same as urinary tract infections, which are UTIs. Okay, so when it comes to fungal infections, these infections, how can we call them? So fungal infections are also known as mycoses. Okay, mycoses. Okay, these which are caused by pathogenic fungi. Okay, so you'll find that we have common types of fungal infections, which include superficial infections such as that which affect the skin, the hair, and the nails. Then we also have systemic infections which affect internal organs, and we also have opportunistic infections which occur in immunocompromised individuals. Okay, so examples of fungal infections, we have what we call athlete's foot, okay, which is also known as tinea pedis. Okay, so this tinea pedis is just a skin infection which is caused by uh, dermatophytes, okay, which lead to itching, redness, and peeling. Okay, then we also have candidiasis. Okay, we also have candidiasis, which is caused by candida species. Okay, candida species, so this can manifest as oral thrush, that is oral cavity infection, or it can also manifest as valvo, valvovaginal candidiasis, which has to do with the yeast infection. Okay, so when it comes to symptoms of fungal infection, symptoms vary depending on the site of infection, but usually we have itching, we have redness, we have swelling, and in severe cases we have systemic symptoms if the infection spreads. Then when it comes to urinary tract infections or UTIs, by definition, UTIs are just infections in any part of the urinary system, including the bladder. So when it has to do with the bladder, we term that or refer that to as cystis. Okay, it can be kidneys, it can be urethra. But most UTIs, it is very important that you understand that they are usually caused by bacteria. Okay, although fungi can also cause UTIs, particularly in immunocompromised patients. Okay, now what is the cause of UTIs? So in most common cases, the causative agents are bacteria like. Okay, the causative agents are bacteria like. So you find that fungal UTIs can occur typically due to candida species, just like I mentioned, in individuals which have a weakened immune system or those with prolonged antibiotic use. Now, what are the symptoms of UTIs? So the common symptoms that we have include a frequent urge of wanting to urinate. So someone who is affected or who is infected with UTIs, you find that they usually want to urinate at several occasions. Then they also have a burning sensation during urination, and not just that, they also have a cloudy or foul-smelling urine. Okay, as well as at some extent tend to have lower abdominal pain. Okay, but in severe cases, there may be even fever and back pain, which can basically indicate renal involvement. So, from what we have said so far with regards to the difference between or in trying to shed light on the what fungal infections and UTIs are all about, we can take down a few differences between fungal infections and UTIs. Okay, so when we focus on the causative agents, you'll find that fungal infections are specifically caused by fungi, while UTIs are primarily caused by bacteria, but can also involve fungi, especially in what? In immunocompromised individuals. Then also the location, as far as differences are concerned, you'll find that fungal infections can occur in various body parts. For instance, skin, internal organs, it can even be oral aspect, but UTIs specifically involve the urinary tract. Okay, and then when it comes to symptoms, that is very easy. We've already elucidated on that matter. Okay, so that was with regards to talking about mycology. Then the other branch of microbiology is parasitology. Okay, so parasitology is a component of microbiology. Okay, what is parasitology? So parasitology is just the study of parasites like worms or malaria causing protozoa. Okay, then we have immunology. Okay, so immunology is just the study of how the body tends to fight diseases. Okay, that is immunology, how the body tends to fight diseases. Okay, that is immunology. Very easy to even understand. All right, so we also have phycology. Okay, we also have phycology as 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 one of the the branches of microbiology. Okay, we also have that now. So phycology by description is just the study of algae, okay, which are primarily aquatic organisms which are capable of conducting or photosynthesis. Okay, very, very important. Remember, algae tend to play a very cardinal role in the ecosystem as the primary producers, thereby tending to contribute to oxygen production and serving as the base of the food web in many aquatic environments. Okay, we proceed now. Let's talk about the historical milestones. Okay, we're talking about historical milestones in microbiology. So we are trying to give a brief history with regards to what has occurred in the field of microbiology from the time people took interest in trying to find out what it is all about. So number one, we are going to focus on early Islamic contributions. So as far as early Islamic contributions are concerned, the first one is um, Ibn Sina. Okay, Ibn Sina. So this particular person actually discovered. Okay, this particular person discovered that some diseases are able to spread from one person to another. So when this person discovered that diseases are able to spread from one person to another, he introduced what we call now as quarantine. Okay, so quarantine just refers to isolation. So why did he introduce this particular aspect of quarantine or isolation? It was with a view of stopping the spread of diseases, because he understood that if somebody is sick, meaning they should be put uh, uh, in a different environment with those who are okay, because they are able to transmit that particular disease or infection to the other person, and hence the disease will spread. Then we also have uh, auras. Okay, we also have auras. So this auras was the first one. Okay, this one was the first one to bring about the difference between uh, smallpox. Okay, between smallpox and basically measles. Okay. Then we also have Al-Zarawi. Okay, we also have Al-Zarawi. So this Al-Zarawi actually, for him, or for this particular person, he wrote medical books. So he wrote medical books regarding infections such as abscesses. So these particular books that the person wrote brought about or showed brought about an early understanding when it comes to microbial diseases. Okay, when it comes to microbial diseases, we continue with the historical milestones in microbiology. So when we let's talk about secondly, we're going to talk about the discovery of microbial life. We're going to talk about the discovery of microbial life. So we have Anthony. Okay, we have Anthony VL. So this particular person was the first one to see living microbes. Okay, so this was the first one to see living microbes. Okay, so living microbes. So that is why we are referring to this particular component of this particular lecture as the discovery of microbial life, because this particular person was the first one to see living microbes. He remember in the in the preamble we mentioned that these microorganisms can only be seen with a microscope and not with our own naked eyes. Okay, so in seeing these living microbes, he used a homemade microscope. Okay, so after he saw those living microbes, what did he do? He called them animalcules. Okay, he called them animalcules. So these animalcules, what that simply means, it just means tiny animals. Okay, tiny animals, that's the name that he gave them. So him being the first one to discover microbial life, or to see living microbes as the first one, he is now considered as the father of microbiology. Thirdly, since this particular person by the name of Anthony brought about the issue of a microscope which was homemade, we cannot run away from the fact of looking at the development of the microscope, even just for a few minutes. Okay, so when it comes to the development of the microscope, we have the person by the name of Zacharias. Okay, so Zacharias made the first, so this one made the first compound microscope. Okay, so the first compound microscope was made by Zacharias. Then Robert Hooke now published a book, micro micrographia. So this micrographia had the drawings of what he saw under the microscope, like mold. Okay. Then we have Galileo and Faber. So these ones helped improve lenses. Okay. So they helped and improve lenses and gave the name microscope. Okay. Number four, we can talk about disproving of uh, the theory known as the spontaneous generation. We can talk about um, the theory of disproving spontaneous generation, because why are we talking about this at this particular lecture? It is because people back then, they used to believe that life, okay, life could actually appear from non-living things. They actually believe that life can appear from non-living things. We call this as abiogenesis. Okay, we call this as abiogenesis. So by calling this as abiogenesis, what that simply means is that what is this abiogenesis? So abiogenesis is just the theory that gives us an idea that life originated from non-living, that is inorganic matter, of which that leads now to the first primitive life form. So this is with regards to abiogenesis. Then we have what we call biogenesis. Remember, bio has to do with the life. Okay, bio is life, genesis is just the beginning. So biogenesis is the idea that life actually arises from the already existing life. Now, let's talk a bit about abiogenesis. Let's consider the history of abiogenesis. Okay, so you find that a few certain microbiologists came on board and took interest, okay, to help prove the concept of abiogenesis wrong. That is to disprove the concept of abiogenesis. So the first one that we have is Francesco Redi. Okay, we have Francesco Redi. So Francesco Redi actually showed that maggots only came from fly eggs, not necessarily from rotting meat. Okay, so this one carried an experiment using to observe where maggots actually came from, and these maggots, it was discovered that they come from fly eggs, not the rotting meat. Then, secondly, we had Lewis. Okay, we had the Lewis. So Lewis, uh, let me take a bit of some time to explain this particular aspect of Lewis's experiment. Okay, so we have the famous swan neck flask experiment. So this famous swan neck flask experiment showed that microbes don't just appear on their own. Okay, so you find that this particular experiment is very important, is or it is a very important demonstration in the field of microbiology. Okay, because it helped debunk the theory of spontaneous generation to the fullest. Okay, so the main aim of this particular experiment, it was the same way as that of Francesco. So what was the aim? The aim was to actually show that microorganisms or microbes are present in the air and can contaminate sterile solutions rather than arising spontaneously from non-living matter. Okay, that is uh, 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 just what Lewis and the famous swan neck flask experiment were all about. Then we also have John Tinado. Okay, we also have John Tinedo. Okay, we also have John Tinedo. So for John Tinedo, he discovered that some bacteria can actually survive boiling. Okay, some bacteria can survive boiling. So he introduced what we call tinadalization. Okay, he introduced what we call tinadalization. So this tinadalization has to do with boiling in stages. So why boiling in stages? It is doing that in order to kill the bacteria which can survive boiling. Okay, so we continue. Why is it very important to actually get to study about abiogenesis? So remember abiogenesis, um, remember that abiogenesis is all about, it tells us that life can come from non-living things. But in the sense, we have the microbiologists that we've talked about, the three microbiologists that we've talked about, which actually contribute to debunking this particular belief or theory. Okay, so studying abiogenesis helps us uh, studying abiogenesis helps us understand how life may have begun on earth. Okay, and also it helps us understand the possibility of life on other planets. So before we conclude with abiogenesis, let's just try to highlight the key differences between abiogenesis and biogenesis. So when it comes to abiogenesis, the origin of life, abiogenesis considers the origin of life to be from non-living matter, while biogenesis uh, tells us that life starts from pre-existing living organisms. Then the founder of abiogenesis actually is Aristotle. Then biogenesis has to do with Lewis. Okay, then, when it comes to scientific support, abiogenesis doesn't have any support, while biogenesis has support, as we have seen from um, the microbiologists who actually contributed. For evidence was there for biogenesis, while abiogenesis was just traditional beliefs or observations. So we continue with our historical milestones in microbiology. Okay, so we can talk about the miasma theory. So this miasma theory is all about the odd view of disease, the odd view of disease. Okay, so according to this particular theory, it is believed that disease actually came from bad air. Okay, came from bad air or vapors from living, from rotting matter rather. Okay, now but this particular theory, the miasma theory, was replaced with the germ theory. Okay, so that leads us to this particular slide, which has to do with the germ theory. Okay, so when it comes to the germ theory, one of the notable microbiologist has to do, is Robert Koch. Okay, Robert Koch. So Robert Koch actually proved that certain or specific bacteria actually cause specific diseases. Okay, such as anthrax, tuberculosis, cholera. You'll find that we have specific bacteria which cause these particular diseases. So the bacteria that causes tuberculosis is not the one that causes anthrax, neither is it the one that causes cholera. That is the germ theory, that a specific bacteria will cause a specific disease. Okay, so under the germ theory, actually, you find that Koch or Robert created the Koch's created the Koch's postulate. So this Koch's postulate is just a method now which is even used even currently to prove that microbes cause diseases. Okay. Then we have something that we call the pathogen pyramid, or the spread of disease, how disease actually is spread. Okay, so you'll find that we have first of all, the individual or the victim may be exposed, and when they are exposed, they'll be infected, and when they are infected, that infection, we are going to have transmission, and when transmission has taken place, we have epidemic spread. The number seven, we have fermentation and pasteurization. Okay, so you'll find that Louis Pasteur, okay, we have Louis Pasteur. So Louis Pasteur proved that microbes are able to bring about fermentation. Okay, for example, making wine, cheese. Okay, so upon him discovering this, or proving this rather, he invented what we call pasteurization. Okay, so pasteurization involves, for instance, gently heating food or drinks. Okay, so why heating food or drinks, for instance milk? It is to kill harmful microbes and make the food or the drink safe for usage or consumption. Then we also have pure culture techniques. Okay, we also have pure culture techniques. So you find that Joseph Lister, okay, so you find that Joseph Lister actually used dilution methods to grow only one type of bacteria at a given time. But then here comes Robert Koch, who made the first solid culture medium using what we call gelatin, of which later he replaced with agar. Okay, and this kind of solid culture medium helped to grow and identify specific bacteria, because it was not like the one that Joseph Lister used, where you can only grow one bacteria type at a time. Then let's also talk about, we are still giving history, let's also talk about medical milestones. So when it comes to medical milestones, um, Gruby and um, Skolin actually okay, found fungi which is able to cause skin diseases like ringworm. Then Pasteur and Koch developed techniques and ideas that these ideas helped revolutionize infection control and disease prevention. Then on number 11, with regards to our historical milestones in microbiology, we can talk about the start of virology, which is the study of viruses. So in 1898, Bell Beijeric, actually proved that viruses, which are usually much smaller than bacteria, can cause diseases. For instance, tobacco mosaic virus in plants. Then now we have Maya. So this Maya actually described the symptoms earlier, but he couldn't isolate the viruses. Now, what is the importance? Okay, what is the importance of us actually uh, uh, or what is the importance of microbiology in medicine? What is the importance of microbiology in medicine? We have it is important in when it comes to disease diagnosis and prevention. Okay, because it helps us identify what causes infections, that is bacteria, viruses, etc. Okay, then it also helps us prevent spread of epidemic and pandemics, because we get to know how diseases work. Then not just that, we also have pharmaceutical discoveries. Okay, we also have pharmaceutical discoveries, and we also have when it comes to biotechnology. Okay, biotechnology. So you find that microbes are used to make insulin for diabetics, clean up oil spills, also engineer crops that resist disease. Then we also have vaccines. Okay, We also have vaccines. Okay, so you'll find that when it comes to vaccines, because we get to know about these microbes, so microbes tend to be weakened or killed in order to make vaccines that help the body build immunity. For example, polio, also recently something that came into manifestation, which has to do with COVID-19 vaccines. Then, lastly but not the least, we also have microbiology is important in medicine when it comes to food and environment safety, because it helps us when it comes to the keeping of food safe by testing for harmful microbes. It also brings about assistance in water purification and preventing pollution. Having said that, thank you very much. This is where this lecture ends.